When humans discovered fire, we explored it, we experimented with it, and then learned how to manipulate it to our liking. This instinct stayed in us, and in 1869, this Swiss biochemist discovered DNA. Then, in 1953, James Watson and Francis Crick discovered the double helix structure of the DNA. Fast forward to 1970, and Gobind Khorana decoded DNA and through the messenger RNA discovers the gene for alanine and synthesized the gene for it in his lab. This was a milestone in our manipulation of DNA. Then by 1981, we synthesized the full gene in lab using Khorana's methods. Then with the invention of PCR in 1983, the field of synthetic genomes was revolutionized. PCR allowed us to amplify and create specific copies of DNA segments, overcoming the issues with handling large strands of DNA. Using PCR, we developed Drexer, which allowed us to replace a section of the genome with synthetic DNA. Then in 2008, we synthesized the complete genome of an organism in lab. This genome had a watermark embedded in it to prove that it was lab-made. This watermark did not hinder the functioning of the genome. Then, later the next year, in 2009, the Gibson protocol was established, facilitating the joining of multiple DNA fractions in a single isothermal reaction. Then in 2019, an artificial genome of Mycoplasma mycoides was transplanted into M. capricolum recipient cell, creating a new Mycoplasma mycoides with only artificial DNA that included a watermark section. From this point onward, this technology was used in making viral coat protein genes to create vaccines, including the mRNA vaccine for the COVID-19. Just like we are advancing in synthetic genomes, we are also advancing scientifically everywhere. So here are some new technologies that aid in the advancement of synthetic genomes. The most recent of these technologies is artificial intelligence. A study in 2021 used two different machine learning algorithms to produce high-quality realistic genomes distinct from the genomes it used to learn. They used generative adversarial networks and restricted Boltzmann machines. These are the two most advanced machine learning algorithms out there. Both of them work by having two computers, one of them that generates genomes and one of them that compares those genomes to real genomes fed to it. Then when the second computer is not able to decipher synthetic genomes from real genomes, the algorithm is advanced enough to create genomes. Of course, this is an oversimplification for the sake of explaining it. Of course, we gotta have data to teach those AIs. There is the National Center for Biotechnology Information, which has 1.9 trillion nucleotide bases coming from 239 million reported sequences. Of course, we have to have a technology to run those AIs on, and this is where the job of supercomputers come in. Supercomputers allow AI to have a playground to train on. Supercomputers allow AI to process these millions and trillions of nucleotide bases and reported sequences. Supercomputers have existed for a few years now, and they are not the cutting edge anymore. Right now, the newest technology is quantum computers. Quantum computers are supercomputers on steroids. What would take AI to practice on supercomputers in a few weeks, it would be a matter of minutes for quantum computers. Yet right now, we haven't yet achieved the ability to fully control quantum computers to use them to teach AI. The future of synthetic genomes could hold the making of hormones in a cheap and efficient way. Right now, we are not at the point where it's cheap or efficient, since 700 kilobases were $200,000 in raw materials. Of course, there are much more efficient ways right now, but in the future, with new innovations, we could end up with something much more cheap and more efficient than what we have right now. It could also be a way to create transplant organs since we can inject DNA into different structures in cells. It has and can be used to create vaccines and might possibly be one day used to create gametes with synthetic DNA that has never existed from any human previously. This, of course, causes questions about the morality of especially the last use, since creating AI generated humans with AI generated genomes is something we have never done before and is something of questionable morality.